Hello. Okay. Hi, welcome everyone. We'll go ahead and get started. Hopefully a few more people will trickle in. So my name is Rachel Logan. I'm a genetic counselor at Children's Healthcare of Atlanta. And I was going to talk today about the very broad topic of genetic counseling. I have some things that I'm happy to share, but my hope always is that it can become a conversation between kind of the questions that you have and how I might be able to answer those. I appreciate the United Leukodystrophy Foundation for inviting me to come talk today. So I'm going to talk a little bit about just kind of some general genetics topics, inheritance patterns. If you've had genetic testing done or a loved one has had genetic testing done, I want to talk a little bit about when you're reading through that report, what are some terms you might find on there and how you might understand them a little better. But the topic of this session is Introduction to Genetic Counseling, so I figured I would do an introduction to genetic counseling. I am a genetic counselor, and what genetic counselors do is we are not physicians, but we are healthcare providers that have graduate level training in medical genetics, and we work with individuals, family members, communities, and other healthcare providers to help empower them to understand how genetic information impacts their lives and the lives of their patients. Some things that a genetic counselor might be able to help you with, or some good questions to ask a genetic counselor that might be involved in your care, would be understanding which genetic testing is being recommended and why. There are lots of different types of genetic tests out there, and so a genetic counselor might help you figure out what is the right genetic test and kind of help inform you as to what that is. So they can also help you understand the results of your genetic testing. So if you've had genetic testing done, or if anyone has gone through that process, they can tell you that there are lots of different types of results we can get back, and they're not always super straightforward. We can help you understand which family members may be at risk for a specific genetic condition. We can help kind of walk you through a decision about genetic testing. So all genetic testing is not right for everyone all the time. And so Julie Cohen is also a genetic counselor that spoke at this conference yesterday, and something she said is, I'm a genetic counselor, I prefer things to be a conversation, and I think that really beautifully describes the work that we do. We prefer to spend our time in conversation and not so much in lecture, but I think this can be a good venue as well. And then helping understand what a genetic diagnosis means in a family. So whether it's yourself or a loved one who's received a genetic diagnosis, what is that going to mean for that person? What's it going to mean for you? And so that's something a genetic counselor can help with. And then we also do education about genetics topics. So what is a gene? So when we talk about genetics or genomics, we're talking about our DNA, our genes. So our DNA is in our body. It's our body's instructions for forming during pregnancy and functioning throughout life. It is packaged into structures called chromosomes. And so in our body, our DNA is contained in chromosomes, and then chromosomes contain DNA. DNA is made up of genes, and that creates proteins. A more kind of a more scientific way of putting this would be, in all of our cells, in almost all of our cells, we have, there is DNA that is contained in them, packaged into structures called chromosomes, and then the instructions for life are contained in 4 chemicals that we abbreviate with letters. And so our body reads that DNA or reads that instructions, it creates a substance called RNA, which then creates proteins, which regulates all of the functions that our body does. Us genetic counselors, we really like analogies. So analogy that I think is really helpful is thinking about our DNA like a cookbook or a recipe. Recipes are things that are passed on through our family members. I know I have some of my grandmother's recipes, and so we get our DNA or our recipe for our body's instructions from our family members, and we keep it in kind of a central part of our home, or like our kitchen. And then our DNA has thousands of different types of recipes to create lots of different types of proteins or foods here. So we get our DNA from our parents. So we get about half of our DNA from our mom and about half from our dad. And that is where our chromosomes come in. So this is what our chromosomes look like. So if we were to break open one of our cells and take all of our DNA and arrange it all neat-like, this is what it would look like. So we have 22 pairs of autosomes. So if you've heard the term autosomal recessive inheritance, that is where that comes from. So it's referring to the numbered chromosomes. When we go to have children, we essentially pass on one of each of our pairs. So we pass on one of our chromosome 1s, one of our chromosome 2s, one of our chromosome 3, etc. And then when we have one copy from each of our parents. For the sake of simplicity, and we know this isn't always the case, but most male individuals have one X chromosome and one Y chromosome. And most female individuals have two X chromosomes. So it's actually typically the dad who determines what the sex chromosomes a baby will have. So for women, we only pass on an X chromosome. And then if that baby inherits an X chromosome from their dad, then they will be typically female. And if they inherit a Y chromosome from their dad, then they will typically be male. As you can see there, there's a little bit of a size difference. The X chromosome is bigger than the Y chromosome. That comes into play. So genetic testing, so for testing for leukodystrophies, when we are noticing signs and symptoms in a patient that they may have a leukodystrophy, there's two main ways that we go about testing. The primary one is genetic testing. So genetic testing is looking specifically at those recipes or instructions and trying to see if there are any differences in those instructions that might cause that gene not to be able to do its job properly. Every gene in our body has a job. And so what we're looking for are misspellings in that recipe. So if we go back to our analogy, that would be like having, extra eggs or no flour or whatever. And so our job then is to say, okay, so if this recipe is missing some pieces or some pieces are spelled differently, would that cause that recipe to not turn out in the way that we would expect? Another way to do it would be to actually look at that process itself in the body. So instead of just looking at the instructions and trying to speculate what the recipe might look like at the end of the recipe, we can actually look at it itself. So that would be things like enzyme testing or specific biomarkers or fats. And that a lot of times these are kind of complementary analysis. So we're looking at the process at two different time points. There are several contexts by which people undergo genetic testing. I think the one that we think about the most would be diagnostic testing. So that's somebody who has symptoms of a leukodystrophy, and so they undergo genetic testing to try to figure out which type it is. But there are other contexts by which people decide to undergo genetic testing. So that can be something like predictive testing, which is kind of like diagnostic testing, but this would be someone who has a genetic condition in their family. They know they're at risk for it, but they're not yet at the age where they've had symptoms. And so they're trying to understand what is their risk to develop that disease. And then there's carrier testing, and then there's something called consultative testing, which is where testing a relative doesn't necessarily have relevance for their own health or care, but it's really more to advise on the genetic context in a family member. So we know that our DNA is shared with family members, and so sometimes testing other family members can be helpful. So maybe kind of preaching to the choir a little bit here, but for some people they ask, well, why would I go about this? If we know that there's something going on and we may not be able to treat it specifically differently, why would we undergo genetic testing? And I think there are several reasons for this. So it can provide a precise diagnosis. We can put a name to it, and there's a lot of benefits to that. It can determine eligibility for therapy. So there are some specific medications where you have to have that precise diagnosis in order to qualify for it. It can help us kind of understand, well, if we know the name of it, then we might be able to learn from other people who have a similar diagnosis. It helped facilitate better research. So on Thursday, we were talking a lot about how knowing that specific genetic variant, the older word for it is mutation, can be helpful in understanding how the disease works. And then it can also provide the opportunity to connect with other people in the community who have the same condition. And I think this is such a beautiful example of that. And then estimating the risk for future pregnancies or for other family members. There are several ways that we go about genetic testing for leukodystrophies. So sometimes I'll have patients come to me and they're like, yes, we would like the genetic test. We would like the DNA to be tested. And you're like, yeah, cool. All right, let's do that. There are lots of different types of genetic tests. And so there are kind of three main categories as relevant to leukodystrophies. So The first example would be single gene testing. So we're looking specifically at one part of the DNA. An example of this would be somebody who has a family member with that condition. We already know what we're looking for. Or somebody's clinical history is just very suggestive of a single disorder. Their physician looks at their symptoms and says, I think it's this, and so we're going to do the testing for that. Another one would be a panel. So there are things called leukodystrophy panels out there. And basically what we do is we take about anywhere between 100 to I've seen as many as 800 genetic conditions that all share something in common. So leukodystrophy can be one of those things as a common feature, but we also have panels for seizures, or we have panels for muscle weakness. And so what it is it's sort of the lab grouping together, what are the most common genetic conditions that kind of all share something in common, and then we can test for those. The nice thing about these is they generally tend to be pretty cost and time effective, and so we can get results sometimes a little bit faster, and sometimes it can cover those particular parts of the DNA a little better. Another way that some people will receive a diagnosis is through exome or genome sequencing. So instead of saying this person has leukodystrophy, we're going to do a leukodystrophy panel. What exome or genome sequencing does, the way that I like to think about it, is we tell the genetics lab about that person's symptoms. So we tell them about our patient. We say, here's what we're seeing. And they look at almost all of their DNA and they report back to us things they think could be relevant for that. And so that is how exome and genome sequencing works. Because it's such a large volume of data, we typically recommend that family members also submit samples. So I work in pediatrics, and so we typically recommend, if both biological parents are around, that we'd like to include samples from them. Not necessarily because we're looking for stuff related to their own health, but rather because understanding the DNA in their family helps us understand how someone's DNA might be a little bit different. And then we talked about this earlier, so there are some tests for leukodystrophies that are more kind of specifically looking at different chemicals or substances in the body which may be elevated or decreased in someone who has that condition. So another context by which people might undergo genetic testing is predictive testing. So again, this is when there's a known genetic diagnosis in the family. The person who is undergoing the testing is at risk for that condition, and then that person is typically younger than the typical age at which somebody might have symptoms for that condition. Something that I, as a gen counselor, do for this sort of thing is we talk about how would we use this information? How have you thought about how this might impact the way that you look at the future, or the way that you look at your own care or family relationships or work relationships? And so there is no right or wrong time, especially if there isn't a specific treatment that we can do ahead of time. There's no right or wrong time to do this. There's some people that feel that all information is good information. They want to know ahead of time. They want to be proactive. And there's some people that say, why would I want that hanging over me? And so I just am going to live my life with the uncertainty that comes with that. That's true for everyone. So what we as genetic counselors do is kind of walk people through that decision-making process. Another form of genetic testing would be carrier testing. So carriers are people who do not have that genetic condition themselves, but they have an increased risk to have a child that has that genetic condition if their partner is also a carrier, or if, in cases for X-linked conditions, if they themselves are a carrier. So carrier testing is much more in kind of the family planning context. It's not typically something that people will do for their own routine healthcare, because for most conditions, being a carrier does not involve any symptoms. For X-linked conditions, that's obviously an exception. But it can be helpful in sort of determining whether somebody has that increased risk and can help for that. And then also there are some contexts which carrier testing is relevant if they're being considered to be a donor for a stem cell transplant, for example. And so that's sometimes when carrier testing can come into play. The best time to do carrier testing is before a pregnancy. So having that information ahead of time allows them to kind of explore all their options. I would encourage you to check out my colleague Julie Cohen's talk about family planning for the leukodystrophies. I believe there's going to be a recording of it available. She did a wonderful overview of that topic. And then in general, carrier testing is something we do in adults. So for most people, they don't have any health risks related to carrier testing. And so typically we recommend, if we know, for example, that there's a child in the family that has a leukodystrophy and they have siblings, and the siblings are at risk to be carriers, we typically recommend that we wait to have that testing done when they're older and they're kind of ready to have that conversation for themselves. So if you're thinking about carrier testing or you've been through it, some good questions to ask your healthcare providers are, is the form of leukodystrophy that I have, does this run in families? Is this something where my other family members may be at risk? And then asking specifically who in my family might be at risk to be a carrier. There are some good resources out there for doing carrier testing. So there are genetic counselors out there called prenatal genetic counselors, and their whole job is to work with people who are planning and building their families, or for people who are already pregnant. So for prenatal genetic counselors, you would sit down with them, you would say, this is something that's running in my family, and I'd like to know if I'm a carrier or if this is something I'm at risk for. I would also like to know, is my partner a carrier? And so there's a findageneticcounselor.org is a good place to find one that's in your community. And then I'm not affiliated with either of these organizations, but they are also out there and have some kind of virtual telehealth options for that. Are there any questions in the chat? Okay. Does anyone have any questions about the genetic testing reasons why people might undergo genetic testing? Okay, yeah. So I'm going to repeat what I understand your question to be, and please tell me if it's an accurate summary. So you have some family members that underwent genetic testing because they have a diagnosis of autism, and because your husband had it. Okay. And then they took some blood for kind of a couple different genetic tests at the same time, and for one of... Got it. So yeah, so if they found something, then they might want to do a more extensive test. So yeah, so I think the root of your question is just kind of getting back to the idea that there are lots of different genetic tests. So there is not like the genetic test that looks for everything. And so sometimes what we'll do is, especially with kids, especially for folks who have sensory sensitivities or on the spectrum, like we know we're going to get one blood draw. And so we try to take advantage of that and think, okay, so if this test is normal, we might want to add something else that wasn't included on that initial analysis. Or sometimes, for example, I think in the example you gave where somebody it's a great segue, by the way, missing a piece of DNA called a deletion. Occasionally it's helpful to know, we can follow that up with, is that the result of a different arrangement of the DNA? And sometimes that involves another genetic test. Also, occasionally we'll start with something like single gene testing or a panel because it tends to come back a little faster. But then if that's normal, we might want to add a more comprehensive genetic test to kind of expand what we're looking for. And DNA in general is a pretty stable molecule. So when we do a blood draw and we have that DNA, we can stick that in a freezer for years. So sometimes what we do is we just kind of try to simplify it for families so they don't have to be driving back and forth to our center for a bunch of stuff. And yeah, if you haven't received the results of that second tier of testing, it could have been because they didn't think you needed it. Could have been, hey, we found it on the first go. We had a contingency plan we didn't need to use. Or it could be that analysis is still pending, and so it'd be worth following up with them to see exactly what that was. It's a good question, thank you. So there are different types of genetic variation. So there's not like one specific type of variant. The older word for that is mutation. So these are kind of the main types that people can have. So a sequence variant would just be related to a different spelling of the DNA. There's lots of different types of those. A deletion is where a portion of DNA is missing. It may involve multiple genes. So it would be like, if we go back to our cookbook or our recipe analogy, it would be like if multiple recipes were missing. A duplication is where there's an extra piece of DNA. So if we go back to our cookbook, that would be like if, 5 recipes or so, there's that. Instead of having it the typical 2 times, they have it 3 times or 4 times. And the body kind of gets confused. They're like, I thought we already had this recipe before. And so that can cause, occasionally, that can cause some problems. And then structural variation is where the DNA is in a different arrangement than it normally is. And so sometimes if somebody has, especially the end of a chromosome missing or something like that, sometimes that can be the result of essentially a different arrangement of the DNA. And sometimes that can inform what the risk for other family members is. And so sometimes we do need to know that. This is one of the more common sequence variants that's out there. So if you have a genetic testing report, you might see language that looks like this. So this is an example of the missense variant. And so on your report, you'll probably see that if it's a sequence variant, you'll see that there's this C dot and then a bunch of numbers and letters. And so what that means is at the 374th position, so if we count all those A's, T's, G's, and C's, we look at all the letters. At the 374th position, most people have a T in this position. But in our patient, what we're seeing here is that they have a C instead. And so our body reads these letters to figure out which amino acids they're going to use for a protein. So amino acids are like the building blocks for proteins. And the way that our body figures out which amino acids to put in order is by reading our DNA. And so in this situation, for a typical person who does not have this condition, and it doesn't matter which one it is, it says the CTG is what tells the body to put a leucine building block on that protein. But for this individual, because that T has been changed to a C, it results in it being a CCG, and so the body puts a proline there instead, which is a different amino acid. For some people, this is tolerated. It doesn't cause any issues for them. And for some people, that would be enough for that protein to look different and to behave in different ways. And so what we do is we always note, this is what we're seeing in the DNA, and then this is what we predict would be resulted in the protein. So that's what that P stands for, is protein. So going back to this, what it says is at the 125th position, where most people would have a leucine, this individual has a proline. And so you can see there on the end, that is what the amino acids actually look like. And so you can see those are different shapes. And so that may cause the protein to behave differently. If you've had any genetic testing past about 2015, you will probably recognize some of these terms from your genetic testing report. So around that time, the American College of Medical Genetics and Genomics decided to standardize the language that we use for genetic variants that shows up on reports. So we all have differences in our DNA. That is the beautiful thing that makes us all different from each other. So there's lots of benign variation in the DNA. That's a good thing. And so what the lab does is they have this sort of reference. They say, this is kind of what's typically normal, with the understanding that there's no such thing as genetic normal, but this is kind of what we typically expect. And every time they notice a difference from that, they will assign every difference that they identify a formal classification. So this is the classification system. So starting at the bottom, that is benign variation. These typically will not show up on your report. The lab knows about them. We as clinicians can ask for them. We don't usually. And so benign variation is normal variation. That's not causing anybody any issues. And so there's strong evidence that this is just normal variation. Likely benign is kind of treated similarly. These also do not show up on your report typically. These are likely normal differences in our DNA. And so these sorts of findings typically will not show up on your genetic testing report. The top 3 categories are ones that will typically show up on a genetic testing report. So starting up at the top, we have pathogenic. And what pathogenic means is that if the right situations occur or the right criteria are met, this variant or this difference in the DNA would cause this person to have this condition. Pathogenic does not mean positive, and I have to explain this to my colleagues all the time. So pathogenic simply means that if the right criteria are met, this would cause this person to have this condition, but we all carry pathogenic variants in our DNA. That's what it means to be a carrier for a lot of things. So pathogenic doesn't always mean that this is what our diagnosis is. So that's on us to help interpret that. Likely pathogenic means that there's very strong evidence that this variant would cause somebody to have that condition. We typically tend to treat likely pathogenic and pathogenic variants similarly. But there's a score that is assigned to every genetic variant that we identify. And so what the likely pathogenic means is that it's really close to having enough criteria met in order to be called pathogenic, but it's just not quite there. We tend to treat those pretty similarly. And then the category of variants that no one likes, I assure you no one likes these, are variants of uncertain significance. And what a variant of uncertain significance is it just represents a knowledge gap. What this tells us is that this particular variation that we found in this person's DNA, we haven't quite seen it in enough healthy people, but we've also never seen it in anybody who has that condition. And so what it likely represents is just a part of our DNA, and we can tell that it's a little different, but we can't tell whether it would actually cause that person to have the condition or not. These results typically are really frustrating. I think the idea is that, you go to these specialists and they're supposed to be able to give you answers, and sometimes we find things that we just don't understand. And on the clinical side of things, it's our job to look at that and try to figure out, does this seem to make sense for what we're seeing in our patient? Or is this something that we think is probably nothing? And with the caveat that we all have variants of uncertain significance, we all have differences in our DNA. And so usually variants of uncertain significance, they do not stay that way forever. It usually just means that we don't know enough about genetics quite yet to be certain about that finding. And sometimes on the clinical side of things, there are things we can do to address that knowledge gap, and sometimes there are things that we just kind of have to wait on scientists to get a little better at. And so if you see that sort of result in your report, a gen counselor is a great person to help you understand what that might mean for you. And then our job is to try to take these 5 categories and squish them down into 3. So we say, our interpretation is this is a positive result, this is a condition that this person has, this is a negative result, even though there might be little things that show up on there, it's not probably causing this person to have this condition. Or sometimes we simply have to acknowledge our own things that we don't know. And we just say this is an uncertain result, we're gonna keep an eye on it, we may recommend some follow-up tests, but it's just at this point, it's just something that we're gonna have to just live with some uncertainty about. And then zygosity, another term that you'll probably see on your genetic testing report is that there are 4 different, generally, zygosities, and this is essentially referring to the context of that variant on the chromosome. So somebody who is heterozygous, which is the one, the green one on the side there, that person has that variant on one of their chromosomes or one copy of that gene, but not on the other one. So that usually implies that the other one looks just fine. Compound heterozygous is where somebody has 2 different genetic variants in each copy of that gene. So for most genes on our autosomes, we have 2 copies of that gene. What compound heterozygous means is that on one copy it's spelled this way, and on one copy it's spelled the other way, but neither of them look like what we would typically see for the spelling of that gene. Homozygous is when somebody has both copies of that gene with the same misspelling. So that can be because that variant is common. It could be because there's some shared ancestry among the parents. But what a homozygous variant just means is that that copy is spelled the same on both in a way that is not typical. Hemizygous is used to refer to when there's essentially only one copy of that chromosome. And so that's normal for our XY male individuals, friends and colleagues. So if we see a variant that's on the X chromosome in an individual who only has one X chromosome, we refer to it as a hemizygous variant because there is not another copy of that chromosome in order to refer to. So that's what, so you will see a variant, you will see the way that it is spelled differently. It's usually an address, it's like a jumbling of numbers and letters, and then you will see what the zygosity is. So is this homozygous? Is it on both copies? Is it heterozygous on one copy? Or you'll see the phrase compound heterozygous if you see two variants in the same gene. So for people who have genetic testing in process, a good question to ask, and this is to the good question you asked earlier, is what genetic test was ordered for me? What did it look for? What did it not look for? Sometimes we can be really specific about what a genetic test looked for. So we can say, we looked for this condition, this person doesn't have it, or they do. There are some things like exome and genome sequencing where we can't really give you a list of all the things that were on there because the list would be really long. So I sometimes will have to tell people, we tested your child for thousands of genetic conditions. The vast majority of them they appear not to have. But I can't necessarily give you a list of all the things that showed up on there because it would just be an untenably long list. And then what does this result mean? If we find something uncertain or we find something positive, does this mean that we are going to do more testing? If it's normal, what tests are we going to do afterwards? Because there is no real right or wrong way to approach genetic testing, and a lot of times it's a conversation between the clinical team and families about what tests we're going to do and at what time. And then if you've already had genetic testing done, what do these results mean? What specific variant was found? If you've undergone genetic testing and you do not have a copy of that report, you should ask for one. I know that people, a lot of times, I know myself, I have this huge folder of just like medical stuff that I keep in a drawer somewhere. That should be in your drawer. A genetic test result is really that kind of original source document that diagnoses somebody with a condition. So if you ever have to prove a diagnosis, a genetic testing document is a good way to do that. It's also a good thing to have for other family members. So if your sister or your brother or your mom is wanting to do genetic testing, a genetic counselor like me is going to need a copy of that report. I have to know what we're looking for. There are thousands of differences in the DNA, so I have to know what it is in order to be able to make sure that I'm offering the right test to your family member. So having a copy of that, having multiple copies of that, having it in an email is an important thing to have. The other thing I just want to mention about the genetic testing process is that it's hard. It's a very emotionally taxing process. It's essentially handing over a very vulnerable question to a process that takes weeks, sometimes months. And for some people who've been on that diagnostic odyssey, it can take years. So I want to just acknowledge that this is a difficult process. And so if you've met with a genetic counselor or you haven't before, our goal is always to try to make that process as clear and as easy as it can be. But because it does take multiple steps, there's always a lot of words people aren't familiar with. And so it really asks for your vulnerability and your trust over a process that can take months. I'm going to go over just some general inheritance patterns. So leukodystrophies can follow multiple inheritance patterns in a family. And then I'm hoping we have some time for questions if people have any. So autosomal dominant inheritance is where somebody only has to have one copy of that gene not working properly in order to have the condition. There's two different ways that this can happen. So in general, this would be a heterozygous variant, if we were paying attention from a former slide. So in this example, this individual's father has the condition, and when he goes to have children, every time he goes to have children, it is a 50-50 chance whether or not that child also has the condition. It doesn't have any preference for like for sons or daughters. It doesn't matter. It is simply that it is 50-50 chance every time. So a question I get a lot is, well, I have one child that has it, so that means the other one will not have it. And that's unfortunately not true. We cannot control the way that our DNA is passed on. It's 50-50 every time. The other thing that also follows autosomal dominance inheritance, so this is relevant for most of the leukodystrophies, would be a de novo variant. And so this is where an individual has one copy of that gene not working properly, the other copy looks okay, but they don't have any parents or siblings that have the condition. And this is where, when that sperm met the egg, it was new in that person. It didn't come from their mom or their dad, and there's nothing that their parents did to control it or to influence it. It's nothing that anybody did wrong. It's just something that happens randomly. We are all born with probably about 5 or 10 new genetic variants, sometimes more, sometimes less. And so it just happens to be, if that part of the DNA is just prone to picking up new variants in the spelling, we have, millions of pieces of DNA and our body has to copy all of those. It would be like if I asked you to take the Encyclopedia Britannica and like hand copy it, you're going to make some errors. And so sometimes that's what a de novo variant is. And it's just something that happens randomly. For that child, when they go to have children, it's that 50-50 again. But a lot of times you can see something that is new in that family and most of the time has a low risk for another child to have it. It's not zero, unfortunately, because sometimes people can carry things like just in their egg cells or just in their sperm cells, which is kind of wild. But in those instances, if both parents test negative, it's usually about a 1, sometimes 2, sometimes higher % chance for another child to have it. So it is pretty low. So autosomal recessive inheritance, this is probably the most common inheritance pattern for leukodystrophies. This is an instance where, in most cases, but not all, both parents are a carrier for that particular type of leukodystrophy. So it's nothing that anybody would have tested you for or you really would have known about. And we all are a carrier for something. It's just whether or not you happen to know what you're a carrier for. And so for people who are carriers, if, in the grand scheme of randomness in life, happen to find a partner who is also a carrier for that condition, then it is a 25% chance every time they have children for that child to have the condition. Presumably there are couples out there who are both carriers for the same thing, who had a couple of kids, who just happen to fall into that 75% chance, and they are blissfully unaware of the fact that they are carriers for that condition. It just is a random thing. For people for most individuals, if a child has a recessive condition, most of the time it means that their parents are a carrier for it. It's worth it to confirm that, just to know for sure, because it can be relevant for other family members. But for most instances, it does mean that they're a carrier. If there is a child who has a recessive leukodystrophy and their parents are both carriers, it usually means that one of their maternal grandparents and one of their paternal grandparents is also a carrier. And again, there's nothing that anybody would have known about. These things can run in families for many generations without anybody knowing. But so for what that would mean is, in this family, this mom's siblings, so if she has full siblings, that means that for each of her brothers and sisters, it's a 50% chance that they are also a carrier. And then for this dad, it is a 50% chance for each of his brothers and sisters for whether they could be a carrier or not. And so for those people, it's important that they be tested and their partner be tested, because then they can know whether they have that chance. So for my unaffected siblings of a child that has a leukodystrophy, for them, if we feel pretty confident they don't have that condition, it is then a 2/3 chance that they are a carrier. So we can see that visually represented here. So if we know that we're in this category of people in light blue shirts, we can see that 2 of them on average will be carriers and 1 of them on average will not be a carrier. And again, good testing to do when people are older and thinking about having kids. Not something we typically recommend in childhood, but just to know that there is that risk. And then X-linked inheritance is always the one that breaks everyone's brain. It's hard for everyone. Even people who work in very closely with conditions with X-linked inheritance, sometimes you're like, hold on, I got to think about it for a minute. And so this is because the whether or not the disease shows up is kind of dependent on the sex chromosomes that person has. So this is an example where mom is a carrier. So in this scenario, our mom has XX chromosomes. She, on one of her X chromosomes, has a copy of that gene not working properly. In the leukodystrophy space, the 2 main ones we see are adrenoleukodystrophy and PLP1, which causes Plesiosmertsbacher, but also some other forms of that condition. Those are the 2 main X-linked ones. So in this instance, mom can have some symptoms as a carrier. They're typically not similar. They're usually different from her male relatives. And so in this situation, when she goes to have children, it's 50-50. So every time, so for a son, it's 50-50 whether or not they have the condition. And then for her daughters, it's 50-50 whether they're a carrier like herself. So anytime someone has kids, it's that 50-50 chance for a boy versus a girl. And then there's that 50-50 chance for her to pass on her X chromosome that has the gene not working properly. And so for dad, when he, I have that on the next slide. So if this is a situation where dad has the condition, so he has on his only X chromosome, he has that gene not working properly, so he will typically have more symptoms of the condition. And for him, when he goes to have children, he will pass on his X chromosome to all of his daughters. So they will all be carriers or heterozygous for that variant. And then, but none of his sons will have the condition because he can only pass on his Y chromosome to his sons. And so that is how that works. Occasionally, we can, with X-linked inheritance, see de novo variants. So similar to autosomal dominant inheritance, occasionally we will see that it is new in that individual. It didn't come from their mom. So if it's a male individual in particular, it didn't come from their mom. It was new in them, and so it is worth checking for the moms of our male individuals who have leukodystrophy. Is worth checking. So that was all the slides I had prepared. I am happy to thank you for coming, especially on a rainy Saturday such as this. And if anyone has any questions, I'm happy to try to answer those. Yes. So for X-linked inheritance. So it depends on the inheritance pattern of the condition. So for an X-linked, so are you talking about in your family in particular? Yeah. So it depends on what the condition is. So if it's something, I think you said it affects chromosome 19. Had adrenoleukodystrophy? Okay, so yes, so for male individuals who have adrenoleukodystrophy, which is X-linked, all of their daughters will be carriers and none of their sons will have the condition. With genetics, there are always some rare exceptions to that rule. It's not, it's very uncommon, but in general, assuming that everybody has the typical sex chromosomes, then yes, if a male individual who has adrenoleukodystrophy, when he goes to have kids, none of his sons will have it and all of his daughters will be carriers. And then for the daughters, and then for somebody who is a woman who knows that they're a carrier for adrenoleukodystrophy, it's 50-50 for all of their kids. So if they have a son, it's 50-50 whether they'll have the condition, and then for a daughter, it's 50-50 whether they will be a carrier. Okay. Yeah, and it's one of those things where when you talk to people who have been impacted by a rare condition, the idea of things just being like, oh, well, you know, it's 50-50 every time or whatever, and it feels like everybody had it or whatever, and it's just random. It's nothing that we can do to really influence it unless you went through something like IVF or like a family planning where you know, did something during pregnancy to, if you know about it, then yeah. Yeah, thank you. Any other questions? Okay. Mm. Yeah. Mm. Yeah. Yes. Mhm. Yeah, no, yeah, I don't. Yeah, so just to repeat what I think you said is that when you went to undergo genetic testing to try to find out about the leukodystrophy in your family, you also found something that you weren't looking for, and that was kind of shocking, and nobody really told you that that was going to happen. As a genetic counselor, that hurts me. Yeah. So especially with exome and genome sequencing, because we can't necessarily we go into the DNA with a question, but sometimes when we get in there, it's a little bit of a Pandora's box, and we can't necessarily unsee what we've seen. And so, in a perfect world with something like exome and genome sequencing, we would sit you down and say, "Hey, I know this is why we're doing this testing. We're hoping that this test will give us an answer, but anytime we're looking at DNA, even if we're being so specific about it, there's a chance we can find things that we weren't looking for or that we're uncertain. And in some instances, you do have a choice about the sort of results you get back. So you can specify a preference. You can say, if there's something out there that's unrelated to why I'm doing this testing, I don't want to know. Like, I am here to focus on the question that I'm here for, and I don't really want to know if there's anything else. And there's some people that say, hey, if we're doing it and there's something else I can know, I want to know. And there's no right or wrong way to think about that. But I think our hope with genetic counseling is that we can try to, have that conversation about, just so you know, this is what this process looks like. And you do have a choice about some preferences you have for things. There's sometimes it's unavoidable, unfortunately. Like because of the nature of the way that we order the testing, it's just, we sometimes find things we're looking for, and we kind of have a duty to report those. And then hopefully somebody would be able to kind of be like, hey, no, this isn't what we were looking for, but here it is. And now that we know about it, we ought to do something. Yeah. Mm-hmm. Right. Mm-hmm. Yeah. No, you read that and you're like, what do you mean likely? Like, right. Right. Yeah. And I think the likely pathogenic criteria got created because we realized that there had to be something in the middle of like, we have no idea and we're absolutely certain. And so the way that is defined in the criteria is that there's a greater than 90% chance that that is in fact a true positive finding. But when you take it in concert with what we're seeing on an MRI and what we see clinically, we can feel very confident. We generally, in the genetic space, tend to treat likely pathogenic variants and pathogenic variants almost the same. Unless, sometimes genetic counselor, I will go back to the lab and be like, I don't agree. And so that is, I think the situation you were in was great, where there was a likely pathogenic variant identified on a genetic test, but your clinicians took that into consideration with what they're seeing in yourself and kind of said, it's likely pathogenic, but it's on here. But I think it is hard for people to see like a sort of a couched finding where it's like, oh, we're pretty sure it's positive. And you're like, what do you mean you're pretty sure? This is a big deal. So yeah, I think that criteria, it's really, I think, just kind of a semantic thing. And it got created just for the sake of being like, well, we need a pretty sure category. And that's where that came from. That's a really good question. I run into that a lot. Well, I'll stick around for another 10 minutes up here if anyone has any other questions you don't necessarily want to answer the group. Thank you for coming. I hope everyone has safe travel home, or if you're from here, safe travels to wherever it takes you. And that's my email. You're always welcome to email me. I'm in Atlanta. Thanks so much.